Good morning. Um, I'm going to be doing a conclusive video on t-test. So if this is the first time you are watching us, I would advise you to watch the previous video that I did on t-test. But today I'm basically uh, teaching you the real application of t-test in pharmaceutical company or possibly in an hospital. And that's why uh, you could see that in my Excel, I have two basic variable. The first one is group variable, and the second one is the test variable. And that's one of the reasons why you should watch the previous video. And when you're talking about the group variable, this is the particular variable in which you will need to define each of the group. As you can see, that the group one is the control. I will show you how to do that, which is the first group, and the second group is the what the treatment, and that's for the first variable, and the second variable is blood pressure. I know you will be thinking that what are we trying to get from this? Number one thing you must know that this is a sample data. Uh, there's a difference between a sample data and a population data. Sample data is just like a particular segment of of individuals or data that is being extract from the major population. So just take note of that. So this is a sample data. So we want to infer from this and conclude if this particular drug can be used in the entire population. So now, why did I call one control? It means that we are trying to confirm or compare the mean of the control drug and the what and the treatment drug. The treatment drug is the drug that is just manufactured that can help reduce blood pressure. So the control is one. The control could just be that oh they are giving them water or they did not even give them any drug. So and they'll be measuring their blood pressure. So but in this case you could see that okay 1, 2, 3, 4, 5. So this is just from 6 entries, possibly from 6 clients, and you cannot just mere looking at it, you can't conclude if the drug is working until you use a statistical measure, you understand, which is inferential, and we are going to be using t-test to know if the drug is effective or not. So the first thing, without wasting much time, the first thing we'll do is that we'll go to our SPSS file. I want to import my data, open from my Excel to my SPSS. Then I think it's under download. I'll go to my download. Come here, go to Excel. Mm, Working hour, no. Control and drug tests. Are you following? Then I'll open. So, okay, now click on OK. So now, as you can see, I've imported the data into my SPSS. Now the next thing is to go to analyze. Then I'll go to compare mean. You can use your t-test under the compare mean. Then just watch the last video, one sample t-test, independent sample. In this case, we're going to be using this independent sample. Independent sample is used when you are dealing with two major groups. The first group is what control I've told you, and the second one is what is treatment. That is the major drug we want to test if it's okay or if it's actually effective in curing high blood pressure. So I'm going to pick independent Now for the group, as you can see, the control is one. So I'm going to put this one to the group part and the blood pressure to the what? To the test variable. Now after that, I'm going to define my group how. The group one is the control, so I'll pick one for control, then two for what? For treatment. Then I'll click on continue. Please don't forget the control is one because I'll be showing you some principles in t-test that will make you know if there is a significant difference between the mean of the two group. So I go to continue, then I'm going to click on OK. Okay, now let's now scroll and see. Now one for control, looking at the mean, you could see that the mean that they are saying is the blood pressure. Can you see? So on the average, the blood pressure of one, which is control, is 140 high, which is higher than the what? The the treatment. That is when they use the drug. Want you to find. Looking at this, you feel there's a difference in the mean, but you can't know until you use what we call t-test for equality of mean, not the Levin test for equality of variance. You want to use the t-test for equality of mean. So now I'm going to quickly tell you how to interpret all this data so that you can know if there is any hypothesis here. You can actually read on what I mean by hypothesis. We have null hypothesis and we have alternate. When you have a null hypothesis, that means there is no even need of carrying out these experiments because there is no significant difference. Well, let's look into this. Now, when you talk about SIG, the two-tailed, not this number one, the two-tailed, is telling us that it's 0.001, 0.0001. Now, I know you'll be wondering what is the meaning of this. So that's what will take me to the principle. To the principle. Let me type the principle so that you follow me and comprehend the meaning. Now, principle one. This principle one is basically for SIG. Now, you must understand something that SIG is the same thing as what? As P what? P value. So the principle says if the P value is what? Is less equal 0.05, it means there is a significant what? Difference between the what? The mean or the what? The average. Hello? So if the mean or if the p value, you understand, and if the p value is greater than, that is greater than what? 0.05, it means no what? Significant. Can you see where they got the sig? No significant difference. No hypothesis. You understand? So now, in this case, let's now go back to this place. In this place, 0.001 definitely is lesser than 0.05. So since it's lesser, it means that there is what? There's an hypothesis, and which means that there is a significant difference. You know, it is when there's a significant difference between the mean of the control and that of the treatment, that is the new grant, the new drug. It simply means that, oh wow, so definitely the drug is what? Is working. The drug is working because there's a what? A significant difference. 0.001 is less than 0.05, which means that there's a significant difference. So when there's a significant difference, you can infer from this that the drug is really, that is the two, as the treatment, the drug is really effective for reducing the blood pressure. Because you can see that by the time they are actually using just water or not even giving their blood pressure is high. All of them has high relative blood pressure. But they are giving some set of people the drug, and by the time we look at the average, you could see that 122, which is reducing, but you cannot still say that, oh, there's a difference until you use this. But this one has shown that there's a what? A significant difference, which means that the drug is actually working. But you can't conclude from this alone. You still need to go and look at what we call the T test, the T value. Sorry, the T value. What is the meaning of this T value? Let me write the the principle again. Principle two. Principle two. Principle two states that if the T value is negative, it means that the group 1 is lesser than the what? Group 2. And if the what? If the T value is positive value, it means that the what? The group 1 is greater than the group 2. So until this one is correct, that's when you can perfect your work and be very sure that it's effective. So now if the T value is negative, but now if the T value is, it means that the group 1 is greater. So let's go and confirm if it's really true. If the T value is positive, and in this case, the T value is positive. Can you see? The T values are positive. Sometimes you see man. So positive means that the group 1 is higher. Can you see that this is correct? Who is group 1? The control is higher. It has an higher mean. So from this, I can tell you 100% with 100% confidence that the what? That the drug is working. And that's why as a data analyst, you don't just conclude based on observation. You use data and different statistical measures to actually give a conclusive answer. I remember I've taken you a lot of topics on statistical measure. We started from the univariance uh, when I was taking you about variance, about standard deviation, about coefficients of variance, and I gave you different real life applications that you can use each of these for. I remember then I told you that you can use it to know the efficiency of a machine. So all these things are used in industry that could really help you. You can use it in trading and in a lot of sectors. So without wasting much time, I think with this, we now understand what is t-test, how to use our t-test to compare mean and to apply it to real life scenarios. I think that is right. Now, something just struck my mind. I know you might be wondering that what is DF? Yeah, you might be wondering that what is what? What is DF? DF means degree of freedom. You don't really need to use this to interpret if there's a significant difference. That's why I only talked about sig, two-tailed, and also T value. This one degree, this one means degree of freedom, and there's a formula that you can actually use to calculate it, which is n1 plus n2 minus 2. You understand? N1 plus N2 minus what? Minus 2. So, but the basic thing about the degree of freedom is that this value affects the shape of t distribution used to calculate significance. So it can actually determine the value that you actually got from what? From your sig two-tailed, but you can't use it to interpret your data at all. So see you in the next class where I will teach you on ANOVA because you can use ANOVA to compare more than two groups. That's just the major difference. So thank you.